Hello, everyone. I'm sure in the last 3 days you have been immersed in the world of lipids, and I'm going to continue the theme of lipids by telling you about how to process the data and analyze the data, and I'm going to suggest you some very interesting tools for analysis of lipidomics data. When we look at our learning objectives for this lecture, will be rudimentary ideas of experimental design. I'm just going to give you a very brief idea or glimpse. I want to tell you how to identify metabolites from MS data. This is covered in the last two talks that you had just now. And then the most important questions that you always come across is how significant is the value of the measured metabolite? Is it statistically significant? So this is an important area I'm going to cover. Factors and differential assessments of significance. You always want to compare two conditions, a tissue with or a cell treated with a drug and without a drug, or a human study between two genders or two genomic backgrounds, genetic backgrounds, etc. So I'll tell you how to look at differential assessment of significance. We'll briefly talk about analysis of mean and variance. We'll look at correlations. Most often, you're interested in trying to ask the question, if you have measured thousands of metabolites or 1,000 metabolites, you want to know how many of them are correlated in a given setting. And we'll address the question of how do you look at correlations. We look at functional significance assessments of metabolites, and then I'll leave metabolites and pathways to the next lecture, which will be on systems biology approaches, and I'll briefly allude to it here at this point. In a metabolomics experiment, you start with a biological tissue or sample, you do extraction of the lipids, and then you take the extraction of the biofluids or material, cellular material, or extracellular material, and then you get biofluids or extracts, which you process through chemical analysis. And I'm going to cover mostly, I'm going to assume that you're looking at mass spectrometric analysis of this sample. And the data will come out in the form of a spectrum, and it'll be something like this. And then you're going to now process this data to make meaningful sense out of this data. The workflow, I'm taking this from Nature Reviews Molecular Cell Biology. The workflow normally has two ways of doing metabolomics or lipidomics experiments. One is called targeted metabolomics, and which is where you have standard metabolites, you do LCMS of standard metabolites, you decide a priori that I know 200 metabolites, I have standards for these metabolites, I'm going to now introduce these standards, and then I'm going to do optimize for the standard, and then I'm going to take my sample, lyse my sample, do LCMS, by now you know what LCMS is, and then we get data, we compare, say two groups, again, processed with reference to the standard metabolite sample so that you get quantification. So the targeted metabolomics often helps you in terms of looking at quantification of your specific metabolites. Untargeted metabolomics is a more discovery tool. You essentially take your tissue, cells, lysates, etc., and then you extract them and you subject them to your standard chromatogram, and you get thousands of peaks and metabolites. You parse them. You will be able to identify some of them. Some of them will be unknowns or unidentified. Then you'll do alignment and analysis, and then you'll try your best to annotate all of those things. And you get a global metabolic profile. If you get a lot of data, you can do relative, profiles, relative values, and so relative quantitation, and you can get some estimate of relative abundance of these metabolites. The Metabolomics Workbench, which is where we have a few thousand studies, typically has data, which is where people submit their metadata and data. And I want you to focus attention on metadata. Metadata is very important because if you want to make comparisons, you need to know exactly what is the type of experiment, what are the experimental factors, and I'm going to tell you what factors are in a minute. And then you'll be able to analyze the data using these factors. In the Metabolomics Workbench, which is, 70% lipids data, because it's all, most metabolites that are measured are lipids. So there are many statistical tools for analysis. I'm going to walk you briefly through the basis of these tools and some examples of these tools. You get normalization and scaling. When you get your data, imagine you're going to compare two different data sets. You want to compare even experiments performed with the same sample at two different times or two different groups by two different groups, or you want to look at comparison between two different samples performed at the same time. You first have to normalize and scale the data so that you compare apples to apples and not apples to oranges. And I'll explain this to you more in detail as we go along. You essentially then convert it to simplistic way of viewing this is the bar graphs and bar plots. You say, okay, I have two conditions. I'm going to do a bar plot and say that in condition B, the value bar plot shows a higher magnitude compared to condition A, for example, etc. So box plots, another way of looking at it. I'll show you examples of those. We can do univariate analysis, Volcano plots, and ANOVA analysis. Very important and very interesting. I'll show you what Volcano plots mean as we go along. You can do class enrichment analysis. We provide something called RefMet, which is like the reference metabolite standards. You can ask the question, is my study going to provide lipid measurements which show that sphingolipids are the highest class of lipids that are expressed or present in my study, for example. You can do clustering and correlation, very much used in lipidomics analysis. You get thousands of lipids. You want to know, for example, can I cluster these into some groups which are certain based on, for example, the quantitative values? Can I cluster them together or by some functional categorization? So we'll tell you a little bit about clustering and correlation. Then your multivariate analysis, very useful tool. In fact, many of you are familiar with principal component analysis. It's a way of reducing your dimensions. Imagine you have 1,000 lipids and you've got 10 different factors or conditions, and you've got essentially a matrix of 1,000 by 10. That's a huge dimension for you to explore to see what is significant. Can we figure out a strategy for us to ask the question, can I find how can I group these into some simple way of classification, classing them with variance as a criterion? And I'll tell you more about it as we go along. Then your classification, feature analysis, things like random forest methods, or PLS-DA, etc. I won't go into great detail, but I'll give you some examples in any case. So first thing I want you to understand before you go further for analysis is the properties of lipidomics data. Average concentration of a single lipid is much lower than the average concentration of a highly abundant lipid. You've got a dynamic range, which means some may be in very small quantities, some lipids will be in very large quantities, which means if you take an average concentration of any given lipid, it may be lower than the average concentration of the most abundant lipid. Imagine in your body, for example, I'm not talking about a lipid. If you take a metabolite, glucose, its concentration is going to be high, right? Or you're going to take some other lipid. For example, in the case of somebody who has metabolic syndrome, your cholesterol values may be high. So it does not mean that the one that is low in amount is not biologically important. This is part of the challenge in analyzing lipidomics data. Second, differences in fold changes in lipid concentrations can vary depending on the roles in metabolism. For example, central metabolism, you may have more of one fold change, fluxing, which means that your substrate is converted into product more, the kinetics is more, and the rate, it's much faster, much higher. And so therefore you're going to convert it. So your flux may be more. So your change in concentrations may vary. And it does not mean that a two-fold change may be more important than a 1.5-fold change. You need to contextualize it with biology. I'll give you examples of that as we go along. Some lipids show large fluctuations in concentrations under this identical experimental conditions. So these are things which are often you see, there's a spelling mistake here, it's conditions. Uninduced biological variation. These are not biologically induced. These are just some random variations due to your experimental error, sample problems and so forth. Technical variations arising from sampling, sample preparation, analysis is most common. And we do try to look for these technical variation by curation methods. And I'm sure you went through MZ-MINE, you went through various other steps which talk about some curation strategies. It is not true that uninduced variations, we talked about uninduced variations, they are not symmetric around the zero or the mean. I mean, they are really can be unsymmetric. They will be more skewed towards highly abundant metabolites as compared to low abundant metabolites. So this is called heteroscedasticity, and this is heteroscedasticity, and this is a very important area. We won't go into great details about this. I'm going to now walk you through a few things in statistics, very simple things, which will help you really process yourself in understanding how we are analyzing this data. Some of these slides were taken from a Canadian bioinformatics workshop lecture of Dr. Shah, and I'm just using it because it was done well, and so I adapted them with credit to him. So for example, how do we describe lipidomics data? So there's always, the data can be varying. For example, the horizontal axis in this plot here, your data goes from small amount to a large amount. The center of data, you want to look at mean of the data, median of the data, mode of the data. I'll tell you what these are in a minute. And you want to look at variability or spread of data. Sometimes the spread is very narrow, which means that the values are all centered around one given value, or it can be large. So the variance, unlike the mean, gives you an assessment of the spread of the data. Then you want to look at relative standing of the data. You want to say, for example, these 20 metabolites are in the highest quantile range of my data, etc. So I'll tell you what that means in just a second. For example, when you look at mean is the average value affected by, the average value is not the central value, is not the median or the mode. The mean is always affected by the tail of the distribution. If the tail is very, very long, then the mean can be skewed towards one side. Median is the middlemost value, usually halfway between the mode and the mean. It's somewhere in between. And mode is the most common value. The most common value you come across is the mode. What should you use? Which value should you use? Depends on the context. Very, very often people use mean, but I'm going to tell you how you adjust the mean to think about these things meaningfully. So variance. Variance is a very important quantity because something can vary between 2 orders of magnitude and something can vary within just the same numerical value, and they are going to be very different in terms of their mean as well as the variance. So variance it's a squared unit change, and it gets increasing contribution from outliers. As I mentioned to you, outliers contribute to variance. Standard deviation is the square root of variance, and I'm going to tell you the formula for it in just a minute. And standard error of mean quantifies the precision of the mean. Takes into account both the standard deviation and the sample size. Very important. Sometimes sample size may be very large, other times sample size may be small. How do you account for that? So you divide by square root of n, which is the sample size. Let's look at mean. Mean is nothing but take all the values for a given metabolite. For example, take the sum of all those values, divide by the number. That's really your mean. Variance is nothing but the given value of ith measurement, subtract from that the average, and then divide by n. Square it, divide by n, and then that's your variance. Standard deviation is the square root of this number. What do you mean by quantiles? Now, often you're used to thinking about this. You say, for example, my data, if I take all my data, break it into the part that is the bottom quartile, which means that just 25%, the median quartile, the top quartile, the third quartile, the fourth quartile, etc. That means it has to be 25% or 50% of the data comes in this, 75% of the data is in this, etc. So this is really an easy way of conceptualizing a large lipidomics data and saying, okay, these classes of lipids fall in this quantile. Okay, now, when you talk about measurements, large measurements, for example, I take, let's say, same metabolite I'm measuring for 20 patients. I'm measuring glucose in 20 patients or cholesterol in 20 patients or 100 patients. I can look at the distribution of the values. And we always think, ideally, that the distribution should be kind of normal, or it has got an average mean, which is kind of symmetric, and then it'll have a width, and the width is often defined by the standard deviation. And this is what we call as a distribution. And we often think about distributions. Almost any set of biological measurements will display some variation, and they will follow a normal distribution. The larger the set of measurements, the more the number, the more normal the curve. If you don't believe me, toss a coin Toss a coin 10 times, you'll get a distribution which is skewed probably. Toss it 1,000 times, toss it 1,000,000 times, you will get a distribution exactly like this distribution like we discussed. Half the time you'll get heads and half the time you'll get tails, and therefore distribution will look like that. Minimum set of measurements, you ask the question, how many measurements should I make to get a normal distribution? There is no magic number to this. I mean, usually if you have less than 30 or 40 measurements, the probability is that you have a skewed distribution. Should we worry about it? No, we'll figure out ways of fixing it as we go along. This is what a normal distribution looks like. This is what one standard deviation looks like, 2 standard deviations, 3 standard deviations, etc. And I'll tell you, which means that you get up to 99% of the data, etc. What about a skewed distribution? Majority of the times, this is ideal, right? You never get normal distribution. You get skewed distribution, which means your distribution will look something like this, where you have a high value at some lower numbers, then you'll have tapers off. Some samples will have low value. It resembles more like a Poisson-like distribution or an exponential distribution. Lots of extreme values from mean or mode. It is not mean or it's not easy to really say that you have got only mean or mode. It's extreme values. It's very difficult to do useful statistics with this distribution. You cannot do meaningful statistics. So what do you do? So what you do is to fix a skewed distribution, for example, it's an exponentially decaying distribution. You can transform it into a normal distribution by applying what is called the log transformation. What log transformation does is to bring the outliers a little closer to the mean, because it rescales the x variable. What does it do? It rescales the x variable, not the y variable, but the x variable. And then it brings it to look like a Gaussian. For example, this is my skewed distribution to begin with. Excuse me for a second. This is my skewed distribution to begin with. And when I log transform it, you can see right away, it goes into a normal-like distribution. You can ask me, why do we care about all of this? How can we apply to metabolomics data or lipidomics data? This is really the question that we are asking. So let's do the following. The first thing you do when you get data is do centering. You convert all the concentrations to fluctuations around 0. That makes life very easy, instead of around the mean of the metabolite concentration. In other words, it adjusts for differences in offset between high abundant and low abundant lipids. So it considers the variation between the samples for analysis. How will you do the centering? I'll show you in a minute. Scaling. Once you center it, divide each variable by a factor, which is different for each variable. This adjusts for differences in the fold differences between different metabolites by converting the data into differences in concentration relative to the scaling factor. What it does is inflates the small values. You can do many types of scaling: auto scaling, Pareto scaling, variable scaling, average scaling. All of this does is normalizes it in some way. And I'll ask you to go over this in my presentation. We are going to make it available to you. I'm not going to go into each of these scaling, except that you, in one case, use a standard deviation, another case, use a square root of standard deviation, in another case, you do a standard deviation and coefficient of variation, etc. Now, this is the formula for doing it. This is taken from this journal paper. You can go and consult this journal paper. This is what it looks like. This is centering. Centering means you're just focusing the unit around 0, focus on the differences and not on similarities of data. You remove the offset, etc. What are the disadvantages? I provide them here. Auto scaling, you've got range scaling, Pareto scaling, variable scaling, level scaling, and these are the methods for doing the scaling. Log transformation, you can also use power transformation. I'm not going to go into details. We don't do it usually in lipidomics experiments. So I'm going to give you a log transformation. Now, how do you know? We do all of these. We get at the end of that, we get a list of data samples. We say these are all the data that we get for lipids. How do you know it is significant? I mean, it may be there, but it may not be significant. To assess significance, we do what is called p-values. P-values provide you a probability that the observed result is obtained by chance. You want to make sure that your result is genuine, and it's not just by random chance you get that particular option. It's just like, again, tossing a coin. You want to make sure that you get a thing by a probability distribution, not by random chance. Usually you set the probability values, p-value, to be less than 0.05 or 0.01 if you want to be more stringent. Sometimes you want to be even more stringent, 0.001. Make sure for multiple testing correction, which means that if you have large number of samples, you want to make sure you normalize with reference to that. So therefore, as a consequence of that, you do something called a false discovery rate. I'll show you more as we go along. If P is less than your alpha, we reject the null hypothesis. That means you've gotten this by random and accept the experimental hypothesis. However, if P is greater than alpha, then we reject the experimental hypothesis that this is significant and accept the null hypothesis that it is not significant. Very simple. So under null hypothesis, all data come from the same distribution. We calculate the statistics, which is mean from the original data. We shuffle the data. The way you do this is, you don't do it, a program does it. Shuffles the data over and over again, recalculates the statistics, and then repeat the steps a zillion times. I mean, actually, you don't do it zillion times, multiple times, and find out where the statistics lies. And this is what it looks like. If this is your threshold, if your observed values here, then none of the permuted mean differences is bigger than the original one to prevent p-value equal to 0. So this is simply a very simple way of computing a p-value. How do you represent p-values in lipidomic data? This is what we do in the metabolomics workbench. We present what are called Volcano plots. This is from Wikipedia. Volcano plots are commonly used to display. What does it do? It essentially plots a log fold change between two conditions as a function of log 10 p-value. And so immediately you recognize it. So therefore, if your value is somewhere in the top level here or top level here, it means it is positive deviation upregulated compared to the control. And here it is downregulated compared to control significantly. Guys which are here in this regime are not significant, so you can ignore them. Very useful at a first glance for you to do lipidomics analysis. I will show you in just 5 minutes how you do it. Then the next question you ask when you do lipidomics experiment is, imagine I have a sample, I have a blood sample taken before treatment with drug A and after treatment with drug A. I want to know, is my lipids change significantly? How do I do it? I can do what's called the paired t-test, which means I can compare the mean between two sample conditions, two samples or two conditions or two features. If two samples are taken from the same population, then they should have similar means, right? You would expect if the distribution or the population is the same, you would expect them to have the same means. If the two means are statistically different, then you know that they are likely to be drawn from two different populations. They are different. So how do you deal with this? So you do what's called a t-test. A parametric t-test is done always with paired sample t-test, and non-parametric test is done with what's called the Mann-Whitney U-test. In the next statistics lecture, in fact, you are going to hear much more about Mann-Whitney U-test and so forth. And these are called the standard student t-tests. So always do a pairwise t-test, and we provide this tool on the Metabolomics workbench. Now, in order to do paired t-test, you first need to reduce your variance. Now, if your variance is very large, for example, a distribution like this, it's going to be, hey, going to be very tough for me to really adjust this. I can reduce the variance by adjusting the variance. And then, for example, this variance is getting converted before and after adjustment of variance, my paired t-tests make more sense. Again, these tools are provided for you on any workbench. I'm going to give you just a couple of more tools before I walk you through examples. Analysis of variance. Imagine I have many factors. I have factor gender, I have genetic background, I have treatment and untreatment, I have time. Say I have 4 factors. Now, what factor is important? Which factor is the p-value that is significant for my lipid? Which factor is not important? So I need to do something called analysis of variance, because if the spread is too much, then I need to know that. So I do what's called multiple group hypothesis testing. H0 means all the means are same. H1 means 2 means are different from others. 1, 2 or more means are different from others. What do we need to do? We need to decide whether the observed difference in means is too large to be the result of random selection. That's a clear goal. So what does ANOVA do? ANOVA is a formal comparison of variation between two groups to variation within the groups. I have 2 featured groups. What's the variation between those compared to the variation within each group? That's what my ANOVA does. I can do, For example, what's called F-test. This diagram illustrates this beautifully. If I take this distribution, this is group 1, group 2, and group 3. Is my variation within this group larger than the variation between these two groups? The answer here is no. So therefore, I can calculate the between group variance and inside the group variance by doing F-test, between group variability, within group variability, and I can do within group variance and between group variance. Very useful tool, because when you do lipidomics experiments, sometimes your experimental condition is such that within a given sample, you'll have much more distribution than before treatment, between treated and untreated, as opposed to between just untreated alone or treated alone. So you need to pay attention to this. So what can be concluded from ANOVA? What you're going to conclude is there significant difference between the groups. Finding exactly where the difference lies requires more statistical analysis. We do more analysis. We do one-way ANOVA. We do factorial ANOVA, mixed design ANOVAs. I won't go into details of this because it's more complicated and sophisticated. But if you want more, maybe the next lecturer on this will go into more details on multi-way ANOVA. Last but important topic, clustering. Clustering is one of the big things that we all do in large-scale omics experiments. We essentially try to take a large number of samples. We try to group them together. Clustering is nothing but grouping. Imagine you have a box of red, green, and blue marbles, and you separate them into red marbles, green marbles, and blue marbles. That's clustering, right? You organize thousands of variables into blocks. Variables in each block are more homogeneous. And this is also a way of dimension reduction, because you're reducing high dimensions into low dimensions. So we do clustering, and it helps you also do principal component analysis, another bread and butter tool for lipidomics measurements. So let's look at this clustering. Clustering comes into many ways. There supervised clustering and unsupervised clustering. Unsupervised clustering means that you don't know a priori any biases. You just say, okay, I'm going to group these according to some measure of Euclidean distance between these things, guys, right? So therefore, I'll walk you through. How do you do this clustering? Agglomerative way of clustering is that you start with one sample, mix them up together, ask the question, one, then kind of combine the two, you get BC, combine these three, you get DE and DEF, combine them. So you essentially start with one sample size of one. At each step of the algorithm, a pair of clusters, the shortest distances are combined. The algorithm stops when all the units are combined into a given cluster. It need not be one cluster. It can be into many clusters if you want. You can specify, I want 5 clusters, for example. What do you need for this? You need the distance measurement between 2 objects and between clusters. That's key for you to decide. Between objects measurements, you can say, okay, I'm going to get Euclidean distance, which means I want to get distances, whatever value, whether it is numerical value of the lipid quantitative value or some other value I specify. For example, I can specify a relative standard deviation or relative mean. I can use any of that to calculate distances, square root of x1 minus xi minus yi squared, summed over all my measurements. So this is called the Euclidean distance. I can do correlation coefficient. It's called Pearson correlation coefficient. The next lecturer will go into detail. I can do Spearman rank correlation, a different type of correlation. This gets me all linear relationship. This gets me a little bit better than that, less sensitive, but more robust. And I can do this clustering. What does clustering do? Each of these different types of clustering, each type of approach, Spearman, Pearson, or Euclidean, gives me slightly different values of clustering. And for example, here, gene A and gene B, gene C and gene D, how they're related to different clustering, they are not the same. They may produce slightly different clustering, clusters. Let's talk about measurement of distance. Single linkage clustering, there are different ways of doing it, complete linkage, average linkage. All that it means is in single linkage, you measure the smallest distance between pairwise distances. You find all pairs, find the smallest distance, right? That's easy to do. Complete linkage is maximum distance of all pairwise distances. Average linkage is average distance of all pairwise distances. You can use any of these. You have the option, depending on the experiment, depending on the distribution of your data, you can use any of these. How do you look at a clustered data? Every one of you has looked at heat maps. People have shown you heat maps. The way heat maps is nothing but a dendrogram. Remember a dendrogram? It's just output of hierarchy. It's a tree. You look at a tree structure and you want to color code the tree structure. The color code looks like this. This is what is a tree. The things on the top and the bottom of the tree. These two guys are closer to each other. Then comes this, etc. So this is the entire tree structure. You represent it as a heat map. Visually, it's very pleasing, visually easy to understand, and so forth. Similarity measurements, how do you do? I already told you, I'm not going to details. Euclidean distance is the absolute measurement of the distance between those two, etc. The last item I want to mention really is principal component analysis. If I have many dimensions, I have 5 features, I have 1,000 metabolites, I have different experimental conditions. It's many dimensions. I cannot deal with all these dimensions. I want to project this high dimensional data into low dimensional data to capture the most variance of the data. So how do I do this? Very easy. I'm going to give you a nice example. This is an example taken from the Canadian Bioinformatics website, lecture website. This is a bagel. If you project a bagel on this dimension, you also get a circle. It looks like this is 2-dimension. A 3-dimensional bagel in 2-dimension looks like this. But if you project it on this dimension axis, it looks like a wiener, a hot dog, right? It looks like a wiener. So it doesn't, because you lose this dimension, you look at this dimension. The O projection captures the most variation. This projection captures the most variation and has the largest eigenvector. And this projection captures the next one, which is, it gives you the length. How long is this bagel, right? That's what it gives you. It gives you the depth. So principal component analysis involves the following. You take this matrix, you do an eigenvalue decomposition of the data, covariant matrix. You have X variables, you have S samples, you essentially project them into scoring them into combinations, linear combinations of these so that they are orthogonal to each other. Very simple idea to the extent possible. So essentially PCA is an orthogonal linear transformation. It's a matrix manipulation problem. So you take the K original variables, produce K new variables, the same number is maintained, but they are linear combinations of your values to minimize variance. So TKs are uncorrelated, unlike X's, unlike these which are correlated to begin with, the T's are uncorrelated. X's are correlated, T's are uncorrelated to the extent possible. T1 explains as much as possible its original variance. T2, the next, usually the way it is T1, T2. If all of them are involved in the variance, then you have not gotten a good principal component decomposition. This is an example of principal component decomposition. I'll show you more as we go along. The principal component analysis can be done with a covariance matrix, which means variables must be in same units, or correlation matrix, which means the variables are standardized. I already showed you how to standardize with correlation. Widely used in metabolomics, you have hundreds of peaks that you get. Your principal component analysis essentially divides them into control samples, PAP samples, ANEAT samples, etc. This is what PCA does, so that if you do your experiment right and if there's real biological change, you see these guys as coming out properly. The loadings, I'm going to skip the loadings, guys, if you don't mind. It's not important. All that loadings tell you is that it tells you the weight, alpha 1, 1, alpha 1, 2, alpha 1, K. These are the loadings. It tells you which variable contributes the most to your principal component. That's all it tells you. I'm going to skip that for this talk. You can go through my slides. Okay. You can do, sometimes principal component analysis does not work when the experimental effects are subtle. So use a method called PLS-DA. It's a supervised method. It essentially is calculated by maximizing the covariance instead of variance. It's covariance between the data matrix and the class labels. It tends to produce certain separations of patterns with regard to original conditions. I'll give you an example. This is my PCA plot. There's no separation. PCA plot, no clear separation. If I do a PLS partial least squares DA plot, I see immediately there is some sort of separation between these things. Essentially, it uses the idea of covariance instead of using the idea of variance. And I showed you what covariance is before. Okay, now use it with caution because sometimes it is based on regression. It first converts the class labels into numbers. So it's susceptible to overfitting. Sometimes you overfit in PLS-DA. So you need to do cross-validation, you need to do permutation tests, etc. Don't worry, all of this is present. Simple statistics package OR. OR is a Bible for statisticians. If you use the OR package, It does all of this for you in the R package, and we do it in the Metabolomics Workbench for you. I'm going to walk you through the next 5 minutes. I'm going to walk you through 5 to 7 minutes. I'm going to walk you through some very simple examples of analysis and tools. Metabolomics Workbench provides you all tools, statistical tools to do analysis. It's www.metabolomicsworkbench.org, O-R-G. And if you go there, you take a sample, you perform data analysis, you can either load your own data or you take the data that is loaded already in the workbench. There are 2,000 studies or something like that. You can load the data, you can normalize and scale. It'll give you an option of normalizing, scaling with Pareto scaling method or variable scaling method. You can do log abundance plots. You can look at bar graphs, bar graph ratios, box plot. You can do univariate analysis all online and download the product of what you're doing. Here's an example, tools for statistical analysis. You can get these PLS-DA plots. I told you this just now. You can get plots like these. And you can get just simply box plots and measurement values of high and low abundance. And this is affected metabolites. This is ceramide 34:1. You can see affected metabolite in female and male, control in female and male, and QC test, quality control test. You can do all of that. You can present them as numerical values with p-values. You can look at heat maps. You can look at a feature and look at heat maps and say, hey, I've got 2 blocks here, right? And then I can look at these values of these, what metabolites they are. Then I can map them to pathways if I want. Here's an example. I'm going to take a few studies to walk you through examples. Here's an example of Parkinson study. We analyzed metabolites of brains of wild type mice and DJ1 knockout mice. The genotype is different. There are 2 genotypes, wild type and DJ1 knockout. DJ1 knockout is a model of an inherited form of Parkinson's disease. So, this is a study funded by National Institute of Aging. They submitted this study. What are the lipids that are different between these 2 studies? We can look at them. I can map the entire thing as a pie chart. I can say, hey, these are all the lipids, metabolites that are different. This is amino acids. This is what we measure, fatty acids, etc. I can look at a pie chart. Beautiful, because I can immediately get a view what's important, what's not, what's abundant, what's not abundant. I can then look at a volcano plot. I told you about a volcano plot. Volcano plot gives me for example, between the 2 genotypes, I want to compare the wild type and the knockout. I want to look at female and male. I want to look at experimental factors or wild type and knockout for female and male, group 1, and wild type and knockout for group 2, female and male. For the knockout, for DJ1 knockout. I can look at a volcano plot of all significant metabolites. I can convert this volcano plot into bar plot. Tells me that these are all downregulated in my given factor. These are all upregulated significantly. For example, a volcano plot looks like this. For instance, I can say, hey, this is very significantly downregulated, very significantly upregulated. Fructose, for example, or anhydroglycitol, for example. So I can then map all of these. I can look at the F value, the P value, the false discovery rate, adjusted P value, look at the class of metabolite, subclass of metabolites. I can look at a PLS-DA plot, beautiful plot, which tells me clearly, separates out those which are upregulated and those which are downregulated. I can map them into VIP scores for my PLS-DA and say that these are all, for example, butyryl carnitine is one of the most important metabolites different between the wild type and the knockout, for example. I could rank order these in order of significance and magnitude. I can look at a heat map. The heat map gives me a very good idea of mapping into different categories between knockout mouse brains and wild type mouse brains. I can look, or male and female, I can look at the differences between what is significant, what is not significant, right? What groupings? It's like, think about it like clustering. So this is, one big cluster, another big cluster, small cluster around here, next smaller cluster, etc. So you can think of it as a clustering idea. I can look at varying experimental factors. Another example, Crohn's disease. This is a study taken from also the Metabolomics Workbench. Crohn's disease, some patient who suffers from irritable bowel syndrome or Crohn's disease, they have control and Crohn's disease. I can look at all the metabolites that are different in a volcano plot, all significant metabolites that are downregulated in Crohn's compared to wild type, all metabolites upregulated in Crohn's compared to wild type. I can do it in a bar plot. I can do it in a plot like this, which says these are down, these are up, etc. I'm going to give you a homework exercise. Now that you've learned what the basic statistics are, go to Metabolomics Workbench. These studies, all these 4 studies have really, we are giving you the output. They are published papers. So we are giving you the output. So you can do your own analysis and then see how well it matches with the output. Okay. This is, for example, the Volcano plot. You can see how it matches with the output for pairwise comparison of 2 experimental conditions, right? This is the bubble plot with reference to, for example, we can also say, find me all the groups of lipids. For example, in this one, lysophosphatidylcholines and phosphatidylcholines are the most downregulated. Sphingomyelins and ceramides are upregulated, etc. So I can look at the size, tells me how many in number are changed. I can do it by class, I can do it by lipid, I can do it by functional category, etc. This is another complex example of Crohn's disease versus control. All the lipid guys and all the non-lipid plots, I can combine them and plot together as a lipid, non-lipid plot. So go home, take all these data sets, take any one of these data sets and this practice so that you get to know how to use these tools for your own pet experiment in lipidomics. Here is again more of the Crohn's disease. For example, these are all the highly changed or differential lipids that are present. N-acetyl, N-acylethanolamines, branched fatty acyls, etc. And then you can look at pathways. This can be translated in the workbench into pathways. For example, these stand for, many of these translate into galactose, fructose, mannose metabolism, carnitine metabolism, endocannabinoid metabolism, etc. So you can map it into pathways. Again, we provide you with tools to look at it in terms of altered metabolite negatively and positively. You can look at them, CD versus control, etc. So please go home and look at it. This is another example from our own study, which we published on NASH, which is a steatohepatitis versus steatosis study. We took many patients, took liver and blood samples, plasma samples, and then did lipidomics. And we showed that, for example, with 20 lipids, we can differentiate between the NASH and NAFLD, non-alcoholic fatty liver disease, steatosis versus NASH. We can discriminate between them with these 20 discriminants. So this is the power of doing statistical analysis in your studies. Last 2 studies, you can do Volcano plots of the same thing. You can look at Volcano plots. You can look at enrichment by metabolite class. You can look at which is down, which is up, etc. These tools are very useful to you. I'm going to leave you by telling you that there's also a tutorial we have, which uses an automated tool called MetEnrichment Package website on the website. If you go to the Metabolomics Workbench and launch MetENP, you can run yourself through any study that you want. You can choose any analysis that you want. You can do, this is what MetENP looks like. You can take this tool, you can use it based on our package. You can look at control samples versus treated samples. You can look at factors, which factor do you want to choose? You can look, this is what your output will, pick any output you want. You want to look at KEGG pathway, MET enrichment score, significance of metabolites, statistics, what statistics you want. You can pick anything you want. It'll give you all these beautiful results. These are results from such a study, visualization plots. It'll give you tables, it'll give you numerical values, it'll give you visualization plots. It'll give you pathways. You can map it into pathways and say, hey, the 2 pathways that are significant here for me are the lysophosphatidylcholine pathways and the ceramide pathways, sphingomyelin pathways. So you can look at pathways and their significance. You can look at bar plots like these on metabolism, which metabolism is important, etc. So I want you to go home and look at this. I'm going to stop at this point. If you have questions, post them on your website. I'm happy to respond to you by email or email me. It's just Shankar, my first name, at UCSD, University of California, San Diego, ucsd.edu. Email me. I'll be very happy to respond to you and help you with your analysis. Good luck. And in the next lecture, I want to walk you through how you can use systems biology approaches to take all the statistical analysis, to translate them into biological mechanisms and biological conclusions and biological phenotypes. This is what I'm going to do in the next lecture. You will have more you'll hear more statistics tomorrow on the next method. Next speaker will talk about statistical methods, more sophisticated methods for statistical analysis. Good luck with the class, and I'm going to come back and talk to you about systems biology of lipidomics.